me know when we're ready to start.
I'm ready.
Going? Okay, so this is the rubber brainstem model, right, that shows us the brainstem, the diencephalon, and also some of our basal nuclei. These are our lentiform nuclei here, which we learned about in class the other day, right, part of our basal nuclei, basal ganglia. This is the internal capsule, which remember we said that was white matter that's going from the thalamus, or the thalami, to the cerebral cortex. Okay, we'll come back to that in a second. These are our thalami. Coming off the inferior surface of the thalami are the medial geniculate nucleus, this bump right there, and the lateral geniculate nucleus, which is where this needle is. Of course, we know the lateral geniculate nucleus is what receives our optic nerve, optic tract fibers, the retinal ganglion cell axons. So they're going to travel here to the LGN, synapse there, upon neuronal cell bodies. These second cell bodies are going to give off their axons, and those axons are the optic radiations. Okay, so the optic radiations, their job is to take the visual information from the LGN back to the primary visual cortex. Holding that thought in mind, let's look at this model now. So, can everybody see? All right. This model shows, well, the thalamus in this model would be deep in this area here, okay? The primary visual cortex is this area in light blue, down there. So you can see there's a problem. Thalamus is here, primary visual cortex is there. The optic radiations can't go straight back because the lateral ventricle is in the way. You can see the same thing on this section here, where the LGN or LGB is this gray matter and its partner on that side. The calcarine fissure here indicates the area where the primary visual cortex is because it lines the calcarine fissure. And then you can see the lateral ventricle is in the way between the two of them. So the LGN here, the calcarine fissure there, the lateral ventricle here. That's going to prevent the optic radiations from going straight back to the primary visual cortex. Okay. So instead what they do is they travel laterally, going around the lateral ventricle and exiting via the internal capsule. See, the lentiform nucleus is right here. This is the caudate nucleus, the thalamus is down there, and this white matter, dog-legged white matter area here is our internal capsule. So these optic radiations are exiting laterally through the most posterior or the retrolentiform portion of the internal capsule, going around the lateral ventricle and back towards the calcarine fissure and primary visual cortex. It's the same thing you can see back on this model again. LGN is where this needle is. This points to the retrolentiform portion of the internal capsule. You see it's behind the lentiform nucleus. The fibers are traveling laterally and exiting from there and then curving around. It's the same thing you can see here. Again, the thalamus is deep. It's going to the primary visual cortex. So you have to imagine the fibers coming out from here and curving around the edge of the lateral ventricle to travel back. And in doing so, they actually make up part of the lateral wall of the lateral ventricle. Now, there is a wrinkle with this, and that's the most inferiorly positioned fibers. These are the ones that are coming off the thalamus, but because they're inferior, they can't travel straight laterally because the temporal horn of the lateral ventricle is in the way. You can see on the ventricular cast here, lateral ventricle is this is the temporal horn. So the fibers, the optic radiations inferiorly, have to travel anteriorly to curve around it. That anterior curving here is called the Meyer's loop. The reason why that's important is because since it is positioned apart from the other optic radiations, if it's injured, or I should say it could be injured by itself while sparing the others. Like, for example, if you had an anterior temporal lobe tumor or stroke or injury or something like that. So it's not uncommon to have a temporal lobe stroke and to get what's called a pie-in-the-sky visual field defect, which is basically a homonymous visual field defect, so it's the same in both eyes, right? It's a contralateral side because we're posterior to the chiasm. It's a superior visual field loss because, as we know, all of the fibers or all that we see is inverted and perverted in the visual system. So basically it would be opposite side, opposite as far as verticality is concerned, and homonymous between the two eyes. It gives kind of a pie wedge-like cut out of the contralateral superior visual field, an injury to the temporal lobe that hits those optic radiations. Okay, so the optic radiations curve around, pass back, go to the primary primary visual cortex. This brings us to the light box here, which shows the presence or a section across the primary and secondary visual cortices. So here the dark material in the center is white matter. The pink material that's lining it at the surface, that is the that's the cortex itself. The cortex has 6 layers throughout. What we can see here is the primary visual cortex has this stripe. You can see it right there. This stripe that's kind of subdividing the pink cortex into 2 divisions. It's not actually doing that, but it looks like it is. That stripe indicates where all the optic radiations are entering the primary visual cortex. Basically, you've got so many myelinated optic radiations entering the cortex here. There's such a high concentration of them, all entering at this one input layer, that it stains more darkly than its surrounding. That also indicates the primary visual cortex, the majority of its function is receiving information. It's receiving all this visual information. It's then going to compute and consolidate all that information down, and then what it gives off, its efferent information, is much less. That's why the secondary visual cortex, which you can see here, does not have a stria like this. This is called the stria of Janéry. It doesn't have a stripe like that because it's not receiving nearly as much information. So this is why the primary visual cortex has a few different names. One of them is Brodmann's area 17, which I think you learned about in your distance assignment. One of them is the V1 cortex, but another name for it is the striate cortex because it has this striped appearance to it when it's stained. And that's the end of our show.
So there's not that stripe right there. It's not on that side.
Right.
Secondary.
Both of these sides would be secondary visual cortex. It's just because of the way this has been cut and all that.
Which is the hostess side, we just start talking.
And what is that point to?
Yeah, this I've saved if anybody has any questions about it specifically. This is a section that would be cut right about there. Okay, through the, actually right about there, just lateral to the LGN. So basically what you're seeing here is the lentiform nucleus there. The lentiform nucleus there. The internal capsule, which is here. This is the retrolentiform portion of the internal capsule, which you can see there. And so that just shows the portion of the internal capsule where the fibers would be exiting.